Today, I'm going to give you a general overview of our technology and our product offering, and hopefully help you see how you can apply it to your application. Our headquarters are located on the University of Colorado Boulder campus in the Biofrontiers Institute building. Not only do we have access to world-class facilities, but some of the best faculty in biochemistry, and arguably one of the best views of the Flatiron Mountains. We all know that scientific microscopes have become indispensable workhorses of laboratories across the world. They are used in applications ranging from single molecule localization microscopy, where one can observe molecular processes beyond the diffraction limit at tens of nanometer, to image cells and tissues that are hundreds of micron thick. But a common theme amongst microscopes is that the objectives they use have been designed to have the best performance at focus. This makes them inherently 2D. One common method of getting 3D information is axial scanning, but that can be impractical for single molecule imaging or tracking. Also, when practical, the limited depth of field of 2D microscopes require finer axial scanning steps, making the process slow. At Double Helix Optics, our technology is enabling researchers to convert their existing microscope systems and techniques to 3D with an extended depth range and best possible precision and resolution without the need for axial scanning. Our technology is widely applicable from nanoscale, creating 3D data down to 25 nanometer of X, Y, and Z precision, all the way to extended depth of field imaging across complete cells. In case you're wondering if you really need Double Helix 3D, let me try and convince you with a couple of examples. Two major applications with Double Helix 3D is 3D single molecule localization microscopy and 3D tracking. In our first example, microtubules from a rat cell are imaged using single molecule localization techniques. This data was collected using STORM, but our technology is agnostic to any kind of single molecule imaging that you do. Shown here are the image on the left is what you would get with Double Helix 3D, whereas the image on the right is what a 2D single molecule microscope would give you. And the bottom two images show the insets as shown in the figure. Here are a few takeaways from this example. Number one, the depth is encoded in color. So as you can see, the Double Helix 3D gave about 2.2 microns of depth of field. And that information has been encoded in colors ranging from red all the way to purple to give you an idea of how the 3D structure of the cell would look like. Number two, extended depth. Clearly, the amount of information that is in the 2D microscope is way less as compared to the Double Helix 3D. So in this case, for example, the 2D microscope gives you about 500 to 800 nanometers of depth of field, whereas with Double Helix, you get about 2.2 microns of depth of field. Not only do we give you extended information, but the fact that you can see the cell in its real 3D information gives you all that structural information back. And then you can take that 3D data and stitch it together to view it from different perspectives like this. So in this particular example, all that 3D data was divided into TIFF stacks and stitched together to give you different perspectives of the same cell so you can study the true nature of the cell. A second major application of our technology is 3D tracking. Double Helix's extended depth allows tracking of particle movement across multiple planes without losing focus. In this particular example, this research group was studying transport processes through nanochannels. This is a very critical step in particle separation, with application ranging from water purification to RNA movement through nuclear pore complexes. To study this, the researchers designed an interconnected 3D structure of hollow glass spheres to observe how particles interact with the surfaces and move across cavities. The image on the left is what you would see when you track this particle in 2D, whereas the image on the right is what you would get with Double Helix 3D. What you notice is with the Double Helix 3D, if you look at the time scale, with white being 0 seconds and red being 180 seconds, it's clear that the particle tends to traverse the entire cavity before moving on to the next one. Whereas when you look at the same data in 2D, clearly all those events moving from one cavity to the other cavity have been misidentified. Not only that, 2D leads to incorrect dwell times, and diffusion coefficients, whereas with double helix is true 3D, you're able to calculate those coefficients as well. Hopefully, I've convinced you that double helix's ability to image in 3D extended depth reveals new insights and information that would have otherwise been lost. Now let's dive deeper into how this is done. A 3D imaging solution consists of three pieces. The first piece is the phase mask library. Phase masks form the core component of our technology and is a secret sauce that enables the efficient conversion of 2D images to 3D information. The second piece of our solution are the spindle and spindle square modules. These are both modular upgrades to your existing microscope systems that make efficient use of our phase mask for optimal performance. The third and final piece is a 3D track software. The software is in the form of an ImageJ plugin and combines the information from your samples as modified by our phase masks and spindle to give you those beautiful and informative images. Next, I'll dive deeper into each of these pieces, followed by a short demo of our software. The first piece of the solution is our patented phase masks that modify the point spread function of your microscope or imaging system. In a conventional microscope, the point spread function is blurred before and after the focal plane. So in this particular example, the image on the top shows you three different points at three different focal depths. The pink point is in focus, whereas the green and blue points are out of focus. If you were to look at the images of those points on a regular 2D system, what you would get is shown at the bottom. So as you can see, the pink point is in focus, whereas the green and blue points are blurred. So clearly, you are spreading your light over more pixels, reducing your signal to noise ratio. Also, the size of the blue and green points are essentially the same because they are symmetric around focus. This makes it so that you can't really identify the Z position of the green point from the blue point. There is an ambiguity in position estimation. However, when we insert a phase mask into the back focal plane of the objective or the pupil plane, this is what you see. Each of those points now look like two lobes that have been separated and have an orientation angle associated with them. And depending on the depth of the point, the orientation angle between those 2 points changes. So as you can see, the amount of light now remains concentrated in a smaller number of pixels throughout the focal depth. And also because it rotates uniformly from one side of focus to the other side, you can extract the 3D information from this data set. I do want to stress that this is all done without axial scanning. So in this particular example, you're simultaneously seeing all three planes on the camera and are able to identify the unique 3D positions by their respective images without the need to axial scan. And as you can imagine, every object is essentially a bunch of points put together. So this same technique can be applied to image the entire objects in 3D. Here is another way of looking at the same example. So in this case, a point emitter is moving in and out of focus. And shown on the left is what the image of that point would look like on a standard microscope. In the middle, we have the double helix. And on the right, we have astigmatic, another popular technique for doing 3D. So what's obvious here is that the double helix 3D, unlike the standard microscope and astigmatic, remains in focus much longer than either of them. The rotating PSF is one example of what we offer. We have a library of phase masks to allow users to match it to their application. Each mask is optimized for precision and depth range, with a depth range extension of about 4 to 30 times that of conventional imaging systems. We even offer multicolor phase masks that show you 2 different PSFs based on the emission wavelength. This allows you to simultaneously image 2 different features of your sample or track 2 differently labeled particles through 3D. The second piece of our solution is our spindle series, specifically the spindle and the SPINDLE². They can both be easily attached to most scientific microscopes and cameras using C-mounts, with most of our users reporting installation under an hour. Phase masks can be easily swapped in and out of the spindle and SPINDLE² to match your application and microscope setup. Each of these modules offer an additional filter slot to further filter out the emission light based on their color or polarization. They both offer bypass modes to return the microscope to conventional 2D operation. Let's dive deeper into each of these now. So for example, here is how you would use a spindle. So typically, you would call us and we would help you select the phase mask matched to your application and setup. The phase mask is then inserted into the phase mask holder and is shipped along with the spindle to let you align it on the microscope. And this whole process, as I said, takes the phase mask matching can take a day or two, whereas the installation takes under an hour. Here is a schematic of the spindle square module. I apologize for the text. Looks like PowerPoint is acting a little funny today. In any case, what we have here is a schematic of the spindle square module. So on the image, you can see that the yellow beam is coming from the microscope to the spindle square module. It is then split into two channels, the green and red channel, either using their emission color, for example, by using a dichroic beam splitter, or using intensity using a regular beam splitter, or its polarization using a polarizing beam splitter. Each channel can be PSF engineered independently or left in bypass modes if that's what the users desire. Additional filter slots exist to further filter out the light based on emission color or polarization. And then both the channels are then combined onto a single camera, making use of the large chip sizes available in today's cameras. If you were wondering how this would look on your setup, here is a quick example. So this is how the spindle square module would look like on a typical scientific microscope. And as you can see, the size of our module is essentially on the order of the camera and smaller compared to a lot of other equipment that you would typically have, showing you how much valuable workspace you can save by using this module. The third part of our integrated solution is a 3D track software to be used with the spindle and spindle square modules. The software has been developed as an ImageJ plugin to shorten the learning curve for most of our life sciences customers. So the way you would use this is you will capture the data using the same tools that you're familiar with on your microscope and import them to 3D tracks for further processing. Our modules, software, and phase masks can be matched to your wide field microscope, your objectives, your cameras, and your wavelengths. We offer a range of phase masks. All of them can span the visible wavelength and even some near IR phase masks. They're easy to install. They allow easy switching between the phase masks. And most importantly, these are passive devices, so there are no electronic parts or no moving parts. They have relatively small footprint. The multi-channel imaging with the multicolor phase masks or the spindle square enables a broad range of biological experiments. They both use a single camera, reducing cost and complexity. And they offer multimodal imaging using different phase masks, polarizers, lenses, or even the bypass mode. Together with this, there are many different applications that you can apply our technology to. Here are a few examples. You can do multicolor 3D single molecule localization microscopy. You can use PALM, STORM, DNA Paint, or any other localization technique for that matter. You can do multicolor 3D particle tracking, multicolor whole cell extended depth of field imaging, SOFI, FRET, and even polarization-based imaging. These modules are additive to wide field microscopes, light sheet microscopes, and even multiphoton microscopes. And I'll show you a few examples of these. Using our technology, our collaborators have enabled new discoveries around the world in life sciences and beyond. We have over 50 publications, and all of these are available on our website. In the next few slides, I will show you a few selected examples from these publications. In this first example, users were trying to study stress granules, which are essential in the study of neurodegenerative diseases like ALS and dementia. Essentially, when a cell is stressed, the cytoplasm clumps up and forms these structures known as stress granules. These granules are on the size of a micron or so. So before super resolution, you couldn't really see any details of the structure. And a major assumption in the field was that these are amorphous structures where the RNA is distributed evenly through the entire stress granule. So on the left, you can see an example of what the stress granules look like with laser scanning confocal. So as you can see, there is no structural data here. It just looks like a blob. They imaged it using SIM, which doubles the resolution, but still could not identify the real structure of the stress granule. Next, they imaged it with single molecule microscopy. And now you can start to see that within this stress granule, there are these core structures where the RNA is concentrated and which become the replication centers for these diseases. But even with that, they could not validate the size, distribution, or even the composition of the stress granules of the core structures. They then imaged it using double helix 3D imaging. And now for the first time, they were able to validate all these three parameters. So again, the size, the composition, and the distribution of the core structures within the stress granules using our solution. I talked about a multicolor phase mask. Let me show you an example of how that would look like in a tracking example. So in this particular data set, there are two different colors. There is, I believe, an Alexa 670 and Alexa 580. And you're trying to image them simultaneously on a single camera. By just looking at their PSFs on a standard microscope, there is absolutely no way of identifying which color is which. However, when we insert our multicolor phase masks, now suddenly you can see that all the green emission colors have a vertical orientation, whereas the red have a horizontal orientation associated with them. So essentially, by using a single phase mask and our spindle module, you can uniquely identify two or more colors on a single camera. Here is what that video would look like. So essentially, those two particles or those two differently colored particles are being tracked in 3D. And as you can see, our software is locating the 3D position. The rotation is what gives you the depth information again, and the lateral displacement of the molecule is what's giving you the XY information. And all that information can be stitched together in videos like this to essentially give you complete 3D multicolor information about your system, or about your sample. Here is another example of multicolor imaging, but this time with a spindle square module. So in this case, the actin was labeled with Alexa 647, whereas the tubulin was labeled with Atto 550 in a COS7 cell. And shown on the right are the images of the two channels separately in their own depth color maps. So here is what the actin looks like, and here is what the tubulin looks like. Of course, to visualize the two different colors together, this would not be very helpful. So one way you can do it is essentially label each of those with a different color, and then you can again view it in different 3D perspectives by stitching the data together. I talked about whole cell super resolution imaging with our technology. Here is another example from Dr. Lee's lab at Cambridge. In this case, they were trying to study how T cell receptors are distributed over the entire surface of the cell. Now, T cells are huge. This cell was about 13 microns or so, I believe. And in order to do single molecule microscopy with that, with a standard microscope, just imagine the number of Z steps you would have to take to be able to get that kind of data. So you would have to do 500 nanometer steps over a 13 micron stack or so. But with the double helix PSF, you could get about 3 microns of data in one single acquisition. So in this case, they did steps of 3 microns, leading them to do only 4 steps to create the distribution of receptors over the entire T cell. Not just that, but they were also able to confirm that the cell is indeed spherical, as shown in this image on the right. And all this data was acquired with a precision of about 10 to 25 nanometers laterally and about 20 to 60 nanometers axially. I had mentioned that you don't need axial scanning, and you still don't need axial scanning. But in this case, this is a very special example where they could combine double helix 3D with axial scanning to even get more depth information. You could do many interesting things with it. So this is just the top of the cell. The video on the top right is showing you the actual point localizations in 3D, and the video on the left is an isosurface created using that point localizations. My last example is from Dr. W.E. Murner's lab at Stanford. In this example, they combined PSF engineering with the double helix PSF and tetrapod with light sheet microscopy. So in this case, they were trying to study a HeLa cell, which is about 6 microns or so in size. And in order to do super resolution and get a huge cell stitched together, they used 2 different phase masks. A double helix phase mask, which was used to collect all the 3D localization data, and a tetrapod phase mask, which was used to track fiducial across Z. And by collecting single molecule data from those 2 different planes and then stitching them together, they were able to generate these entire whole cell images in super resolution. What's really interesting about this is when you think about microscopes, either they give you high resolution data or they give you whole cell data. This is a very unique example where you could combine both of them. And if you look at the image on the bottom right, you can see the tubular structure of the mitochondria as well. And on the left side, you can see the whole cell at the same time. I do want to point out that we offer both these phase masks and our multicolor masks, whereas the SPINDLE², also the SPINDLE² module, can allow you to capture data like this if you have a light sheet microscope. Thank you for your attention. I hope I've convinced you that a double helix 3D solution enabled by a library of light engineering phase masks enables you to see like no other.